from which all other life activities are fueled. So similarly, as we know, we cannot make use of this solar energy, and plants, which are the producers, they make use of this solar energy, and they capture this energy in the bonds, in the chemical bonds of substances, say for example glucose. So what we actually see is a reversal, is actually a reversal of photosynthesis. Whatever cellular respiration we see, this is actually reversal of photosynthesis, wherein photosynthesis we make use of light energy, and we build up glucose, which is energy rich molecule. We infuse energy to energy deficient molecules like water and carbon dioxide. We infuse energy to these molecules, and we synthesize carbohydrates. The energy is there in the bonds which make these molecules. First slide, Monica. So this consuming these biomolecules,
Is it visible?
Yeah. So making use of these molecules which are synthesized by the producers, the consumers get these molecules from the producers, and they burn this. Actually, it is called as physiological combustion. Combustion is nothing but burning. As we burn firewood to get heat and light, it's similarly the same process, but in a very conserved environment. And this conserved environment, by this I mean, see, if you burn a pile of firewood, you will get enormous amount of heat, energy in the form of heat. But in our body, if we burn glucose, we can't have that drastic conditions. So what we have is we have the restrictions of temperature and pH. So this is looked after by enzymes. I hope people are following what I'm saying. What we do is we just burn the biomolecules, say for example glucose, in our body without exploiting full energy or extracting full energy, complete energy of that molecule, without increasing the temperature or pH with the help of enzymes. So ultimately what happens is we store all this energy in the form of our energy currency, which is ATP. This ATP is used for our all other physiological activities. Next slide, Monica, please. So this was what I told now. I just told you now. The primary source of all the energy that we get, that we have in a planet, is from the sun, and that is used by the producers, which are autotrophic organisms of plants, which comprises of plants, algae, and some bacteria, which usually does photosynthesis. Consumers are heterotrophs, which depend primarily on the producers or the autotrophs, which comprise of animals, fungi, and other protists, which is bacteria. They make use of cellular respiration. This is nothing but physiological combustion, wherein we burn with the help of oxygen, and we extract the energy. So ultimately, the energy is stored in the form of ATP, which is adenosine triphosphate, our energy currency, which is used to store and release energy when it is required. All the physiological activities of the body are dependent on ATP. Next slide, please.
It has changed.
General reaction of this is like this. We burn a molecule of glucose with the help of 6 oxygen molecules to release 6 molecules of carbon dioxide and 6 molecules of water, releasing energy. And what we generally see is we measure energy in terms of calorie. Can anybody say what's calorie? What is calorie?
Yes, anyone is
Hello?
there? You people know what is calorie, right?
Hello?
Yes, yes, you are audible. You are audible.
Sir, it is the unit of joule, unit of energy. Am I audible, sir?
Yes, yes, yes.
Hello?
Yeah, you are audible. Yes, yes,
It is
Yeah.
the
Yeah. Huh. Hello?
yeah, it is the unit of energy.
Unit of energy, exactly, which is used to increase the temperature of 1 centimeter cube of water. Okay, and you should also know how much that is in joules. Okay, that is the unit of energy which we usually make use in food and nutrition area. So this is about it. Then going to energy harvesting, we have three important biological pathways or metabolic pathways employing which we extract the energy from glucose. I say glucose mainly because next slide. Next slide. As Mausami has already told, around 70% of our diet is mainly made up of carbohydrates, and these carbohydrates ultimately end up being glucose. The food which we eat are generally made up of pulses and cereals, and cereals are the ones which are made up of which have starch in them. Say, for example, wheat, rice, ragi, and all these cereals. They have starch as their main component, and this starch is digested with the help of amylase, and that releases glucose. Also, we have other enzymes in the intestine. Say, for example, maltase, um, all these enzymes, which convert other oligosaccharides also into glucose. Ultimately, this glucose is absorbed in the intestine and it reaches the bloodstream. After reaching the bloodstream, this is delivered to all the parts of the body, all the tissues in the body, and each tissue has to burn this glucose to extract energy. And what we see now is the cellular respiration generally, which takes place in three important steps. First is glycolysis. As the name suggests, glyco means sugar, lysis is breakdown of sugar. What we generally see in glycolysis is we break down one glucose into two molecules of pyruvate. Glucose is a six carbon containing molecule, and pyruvate is a 3 carbon containing molecule. We break one glucose into two pyruvate. Later, this pyruvate is transported into the powerhouse of the cell, that is our mitochondria, where pyruvate is converted into acetyl CoA with the help of pyruvate dehydrogenase complex, which is there in the mitochondrial matrix. Pyruvate is converted to acetyl CoA. Acetyl CoA enters the Krebs cycle or tricarboxylic acid cycle, and it generates reducing power. By reducing power, we mean NADH and FADH2. So these are further sent to electron transport chain. Electron transport chain is an array of oxidoreductase enzyme complexes. They are usually four: complex 1, complex 2, complex 3, complex 4. By passing the electrons which have the energy in them in these complexes, we make a proton gradient. I'll explain in detail in the next coming slides. At the end of this electron transport chain, we will be able to synthesize ATP. So this is the overall picture of our cellular respiration, which starts with glycolysis and ends in the electron transport chain by generating ATP. Uh, what we are looking at is we are extracting electrons from the biomolecules, from all the intermediates starting from glucose. We are oxidizing this, and the ultimate electron acceptor, energy rich electron, is made to pass through an array of uh, complexes at different energy levels. As the electron comes down from one intermediate to another intermediate, the energy comes decreasing. Ultimately, when all the energy is extracted, energy deficient electrons will be ultimately absorbed by the oxygen, converting oxygen into water. Okay, this is exactly the reversal of photosynthesis. What we saw, photosynthesis was carbon dioxide plus water gives us glucose and oxygen. Here, what we do is we combine glucose and oxygen to give out water and carbon dioxide. Am I clear? Are you people are able to follow me?
Yes, it's very clear. Yes.
Okay. Next slide, please.
Okay.
This is what I told you. We generally break one molecule of glucose into two molecules of pyruvate in the cytoplasm. Glycolysis occurs in that cytoplasm, and this glycolysis is independent of oxygen. Oxygen is not required. And when there is anaerobic condition or oxygen deficiency, our cells have an alternate pathway. That is the fermentation fermentation, wherein we convert pyruvate into lactic acid. Next slide, please. Coming to the steps of glycolysis. Next slide, please.
I have changed the slide. Is it not visible? Yes.
Coming to the steps of glycolysis, we start with glucose, which is the 6-carbon molecule, which is a basic hexose, aldohexose, and this is converted to glucose 6-phosphate, and all the other intermediates up to pyruvate. All the other intermediates up to pyruvate are phosphorylated. You can see everything has a phosphate group. This is very essential because whatever is phosphorylated cannot simply escape the cell. If glucose is present as such in the form of glucose, glucose can diffuse in and out of the cell easily. But once it is phosphorylated, glucose is bound to go into the further steps of glycolysis. So the first step is pushing glucose into this pathway, that is converting glucose into glucose 6-phosphate. Here we consume one molecule of ATP, and the enzyme here is hexokinase. This hexokinase is present in all the parts of our body, and a similar enzyme or an isoenzyme of hexokinase called glucokinase is present in the liver, which works only at higher concentrations of glucose, which we find in the liver. Next, after glucose is phosphorylated to glucose 6-phosphate, it is isomerized to fructose 6-phosphate. This fructose 6-phosphate is an isomer, and this is an so this is a hexoketose. It's a ketose sugar which has a C double bond or carbonyl group. Again, this is phosphorylated with the help of one more ATP, generating fructose 1,6 bisphosphate, which has phosphate group attached both at first and sixth carbon atom. These two steps, or these two enzymes, hexokinase and phosphofructose, since they make use of ATP, these are irreversible. These two steps, conversion of glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate, both are irreversible steps, and all others except the last one, conversion of phosphoenol pyruvate and pyruvate, all other steps are reversible. Then once we get fructose 1,6 bisphosphate, this is acted upon by an enzyme called as aldolase, breaking down it into one molecule of DHAP and one molecule of glyceraldehyde 3-phosphate. Again, these two are isomers. One is an aldotriose and the other one is a ketotriose. Glyceraldehyde 3-phosphate is an aldotriose. Dihydroxyacetone phosphate is a ketotriose. It's a ketose containing 3 carbon atoms. These two are again one can be converted to other with the help of triose phosphate isomerase enzyme. Once we get glyceraldehyde phosphate, one more important thing in this step is glyceraldehyde 3-phosphate is preferred if the cell is going for glucose utilization, or if we have plenty of glucose and if we want to store this in the form of triacyl glycerols, DHAP is preferred, and DHAP is taken and converted to glycerol, and this glycerol is used for making triacyl glycerols. This is the thing. So if the cell wants to go for burning glucose, even the DHAP is converted to glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. Again, this step is an ATP consuming step, but this is reversible. After we get 1,3-bisphosphoglycerate, that is three carbon containing molecule with phosphate groups both on first and third carbons, it is converted to 3-phosphoglycerate by generating one ATP. Here in this step, we take up the phosphate group from 1,3-bisphosphoglycerate and prime it with the ADP to make one ATP. And in the next step, we have a mutase reaction, phosphoglyceromutase, which shifts the phosphate group from third carbon to the second carbon molecule, second carbon atom. So the next step is enolase step, wherein 2-phosphoglycerate is converted to phosphoenol pyruvate, wherein there is a conversion of aldose group to keto group or enol group, and phosphoenol pyruvate. In this step again, we take up the phosphate group from the PEP or phosphoenol pyruvate and prime it with ADP to generate one ATP, and ultimately we get one pyruvate. I hope this is clear. Now coming to the calculations of energy, how much we have spent and how much we have got. In the first step, we have consumed 1. In the hexokinase step, we have consumed 1 ATP. And in the phosphofructokinase PFK step, we have consumed 1 ATP. But we have generated 4 ATPs in the subsequent step, because 1 glucose yields 2 glyceraldehyde 3-phosphate. The 2 ATPs generated in the phosphoglycerokinase and pyruvate kinase steps are considered into 2 into 2, 4. Then we generate one NADH in the step where we convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. In that step, glyceraldehyde 3-phosphate dehydrogenase step, we generate one NADH. This NADH has to be oxidized back to NAD plus if the glycolysis has to continue. If there is absence of oxygen and if the glycolysis step has to take place, the NADH which we generate in that step has to be oxidized back to NAD plus. This is done by converting pyruvate into lactate. In the process, NADH will be converted to NAD plus again, and the glycolysis can continue without the help of oxygen. This was about glycolysis. Is everyone able to follow? Hello. Hello.
Yes,
Okay.
sir.
Yes, sir.
Yeah,
Yes.
next coming to Krebs cycle. The pyruvate which we generated in the glycolysis enters the mitochondrial matrix. Here we have a huge enzyme complex that is called as pyruvate dehydrogenase. This converts pyruvate to acetyl CoA. In the step, it generates one NADH again. This acetyl CoA will is primed with oxaloacetic acid, which is a 4-carbon molecule. Acetyl CoA is 2-carbon molecule. These two combine with the help of citrate synthase enzyme, generating citric acid or citrate. This is a tricarboxylic acid cycle because, as you can see from the slide, next slide, please. Sorry, previous slide.
This one.
Yeah, this one. As you can see, all the 3 carbon atoms on the backbone have the carboxyl group or COO group. So this is called as tricarboxylic acid. Since citrate is a tricarboxylic acid and the first member of this metabolic pathway, this is also called as tricarboxylic acid cycle. And this pathway was discovered by the scientist Hans Krebs. So in commemorating his memory, we have named this cycle as Krebs cycle. So once we get citrate is isomerized into isocitrate, and isocitrate is converted to cis-aconitate, which is not shown here, and that will be oxidized, and this is called as oxidative decarboxylation. Very important. This is the step where we get our first carbon dioxide molecule. So we have the 6-carbon molecule isocitrate, and from that we are removing 1 carbon dioxide and getting 5-carbon compound alpha ketoglutarate by generating 1 NADH. In the subsequent step, again we have one more oxidative decarboxylation of alpha ketoglutarate to succinyl CoA, and this succinyl CoA is a high energy compound. Wherever there is CoA and there is breakdown of that SH bond, sulfhydryl bond, there will be generation of energy. This succinyl CoA will generate succinate by giving off coenzyme A, and in the step we generate one GTP, and this GTP is utilized for generating one ATP. This is one ATP generating step of the Krebs cycle, and subsequently this succinate is converted into fumarate. In this step, we generate FADH2. This is important because this FADH2 is the substrate for second enzyme complex in the electron transport chain. We will see that later. And succinate will be converted to fumarate. What we do is simply we remove the hydrogen or we oxidize it. Then fumarate is hydrolyzed or added with one water molecule to get malic acid, which has one OH group. This malic acid is again converted to oxidized to generate NADH. Is oxidized to generate NADH, and the resulting keto form is called as the oxaloacetate. So we started with oxaloacetate. What happened is we have oxidatively decarboxylated citrate to get back oxaloacetate. Whatever two carbon atoms which enters in the form of acetyl CoA is lost in the cycle to give 2 carbon dioxide. Meanwhile, we get 2 NADH. 3 NADH, one in the step of converting isocitrate to alpha ketoglutarate, then alpha ketoglutarate to succinyl CoA, and from converting malate to oxaloacetate. In these 3 steps, we generate NADH, and succinate to fumarate gives us one FADH2. All these are called as reducing powers. These will be further oxidized in the electron transport chain to generate ATP. Also, conversion of succinyl CoA to succinate generates one ATP. Krebs cycle is called as amphibolic pathway. This is very, very important because amphibolic pathway says that this has the nature of both anabolic pathway and a catabolic pathway, and especially alpha ketoglutarate, oxaloacetate. These can be taken away from this pathway to generate amino acids. These are the important intermediates for generating amino acids. And citric acid is shuttled to the cytoplasm, and it generates acetyl-CoA there, which leads to the formation of biosynthesis of fatty acids. Citrate is the precursor which enters from mitochondria, where this Krebs cycle is taking place. It goes to cytoplasm, and it gets converted to acetyl-CoA, the precursor for biosynthesis of fatty acids. So in this way, we take away intermediates to generate other important biomolecules. Also, there is replenishing of intermediates, that is called as anaplerosis. Anaplerosis is nothing but we give out various intermediates to keep the cycle continued. So as we can see, amino acids in the protein metabolism we study, our glutamine and glutarate are converted into alpha ketoglutarate, which can enter the Krebs cycle. Similarly, we have aspartic acid, which can be converted to oxaloacetate, and various other important things will also be replenished. And one more important thing is succinyl-CoA is used for is taken away for synthesizing heme of hemoglobin and cytochromes. This is one important information. This Krebs cycle is a very central pathway in the metabolism because this is amphibolic, being both catabolic and anabolic in nature. So next slide, please. Next, moving on to electron transport chain. As we have seen in the Krebs cycle and also in the glycolysis, we have the reducing powers with us now, and these reducing powers have the energy-rich electrons in them. So what we do is we sequentially dehydrogenate these reducing powers to get energy-rich electrons. These energy-rich electrons give out the energy, and this energy is used for pumping out the proton from inside to the outside of the inner mitochondrial membrane. So these are the enzyme complexes 1, 2, 3, and 4 are found in the inner mitochondrial membrane of the cell. As we all know, mitochondria is a bilayered structure. It has two membranes, outer membrane and inner membrane, and there is a space between these two membranes that is called as intermitochondrial intermembrane space, and we have the matrix inside the inner mitochondrial membrane. NADH, which is generated from the tricarboxylic acid cycle happening inside the matrix of the mitochondria, are taken up by the complex 1. Complex 1 is called as NADH dehydrogenase complex. This is made up of flavoproteins, iron sulfur complexes, and it is made up of these molecules. This is a complex of many proteins and other important coenzymes. So what it does is it accepts NADH as its substrate. It oxidizes it to NADH. In the process, whatever proton is given out, it is pushed outside, utilizing the energy in the electron. So in this picture, we have shown only complex 1. Complex 2 is not shown. Similar to the complex 1, complex 2 accepts FADH2, which is generated in the succinate to fumarate step of Krebs cycle. That FADH2 is oxidized by the second complex, second ETC chain complex, which is called as FADH dehydrogenase, and it also pumps protons outside. There is one important coenzyme called as coenzyme soluble enzyme on molecule, which is a mobile transporter. So this moves between 1, 2, and 3. The coenzyme Q accepts electrons from the first and second complexes, and it gives these electrons to the third important complex of the electron transport chain. Third complex is made up of cytochromes, cytochrome BH, BL, and cytochrome C. This accepts electrons from the coenzyme Q and passes those electrons to cytochrome C. During this passage of electrons, the energy of the electrons is harvested to again create a proton gradient, which pumps protons to the intermembrane space. And then we have a cytochrome C, which is the electron acceptor from the complex 3, and it gives out this to complex 4, which is called as cytochrome A, which is made up of cytochrome A. This complex ultimately reduces the oxygen, giving out water molecule. All these protons which are pumped outside actually result in increase of pH in the mitochondrial matrix. Since protons are pumped outside, the pH increases in the matrix and pH decreases in the intermembrane space. And also there is a difference in the electrical charge. So there is a higher positive charge in the intermembrane space. So this is called as the electrochemical gradient. So making use of this electrochemical gradient, gradient pushes the proton back to the matrix. So this is utilized by one more important complex that is called as ATP synthase, or F0 F1 ATP synthase, which is again an important intermembrane protein complex, and this lets in the protons in the direction of gradient. That is, from where there is higher concentration of protein, it lets the proton to come to the region where there is a deficiency of proton. So when such movement along the gradient happens, there is utilization of energy, and that energy is harvested in the form of ATP production. So making use of that pushing energy, ATP is synthesized in the F1 F0 complex. So last step is called as oxidative phosphorylation, because we are ultimately oxidizing glucose into pyruvate into acetyl-CoA. Meanwhile, we generate NADH, and NADH are ultimately reduced. These reducing powers are also again oxidized. Everywhere we are oxidizing everything. By oxidizing, we are getting the ADP phosphorylated, or addition of phosphate group. So this is called as oxidative phosphorylation. There are actually 3 important types of energy transfer reactions which are associated with phosphorylation. First is photophosphorylation, which we can see in the photosynthesis. In photosynthesis, we have the light harvesting complex, and we use the light energy to produce ATP. That is called as photophosphorylation. The second one is this, which we see in the electron transport chain. This is oxidative phosphorylation. And the third one is, say for example, conversion of glucose to conversion of phosphoenol pyruvate to pyruvate. In that step, phosphoenol pyruvate phosphorylates ADP to give out ATP. So this is again a important phosphorylation, but this is substrate level phosphorylation. These are the 3 important types of energy transfer reactions involving phosphorylation. One is photophosphorylation in photosynthesis. Second is oxidative phosphorylation in respiration, and the other one is phosphorylation of the intermediates of any pathway, which is called as substrate level phosphorylation. Next slide, please. So here, for an example, how proton is pumped outside, I have taken the example of a Q cycle. Q cycle is nothing but how the coenzyme Q works. So what we see here is the complex 3, which is made up of cytochrome C1 and cytochrome B, L, BH, and cytochrome C1. These are the important proteins which make up this complex. In this complex, as you have to observe the diagram, there will be on the left side image, you can see two coenzyme Q molecules. One is QH2 on the top, which is in the yellow box. QH2 is the reduced form of coenzyme Q that enters the complex 3, and one more molecule of non-reduced form of Q, coenzyme Q, also enters. What happens is the electrons, will be given from the complex complex 1 and 2. Electrons taken from complex 1 and 2 will be brought to the complex 3 in the form of reduced coenzyme Q, that is QH2. This QH2 gives electrons to iron sulfur protein, which is written as 2Fe2S, iron sulfur protein. That in turn gives electrons to one electron to cytochrome C. This cytochrome C ultimately passes the electron to cytochrome C, which in turn goes to complex 4. And one more electron is given back to non-reduced coenzyme Q. This coenzyme Q becomes ubiquinone radical, free radical. It takes up one single electron and becomes a superoxide radical. In the next step, again, when one more reduced ubiquinone comes from the complex 1 or 2, at that time, this superoxide radical takes up the protons from the matrix, and it pumps out the protons outside the out to the intermembrane space. So this is how proton pumping works. So these intermediary molecules, they are actually oxidoreductases. They take up electrons from something and they give out electrons to some other thing. Meanwhile, energy is harvested to create a proton gradient. This is what we see. Both are half cycles. We call it as one half cycle of Q cycle and the other half cycle of Q cycle. In the first, we pump out 2 protons and 1 electron and generate one superoxide radical of ubiquinone. In the second half cycle, we again reduce that to QH2 and also pump 1 electron outside and 2 electron to cytochrome C and 2 protons to the intermembrane space. So on the right side, we have the image of F0 F1 ATP synthase. This is an important multi-protein complex, which is made up of 2 important subunits. The one which is anchored in the membrane, which you can see, that is called as the F0 part. The other one, the club-like structure, which is protruding towards the matrix, is the F1 complex. F1 is made up of 3 repetitive subunits of alpha and beta subunits, which has